Hello, my name is Smilee Samuel, and I work as the project coordinator at the CLINS section for the Office of Cyber Infrastructure and Computational Biosciences with NIAID. So here at NIAID, the BCBB team basically is helpful in driving the innovation for bioinformatics information for global health clinicians and researchers by providing a suite of products, platforms, and solutions, along with educating them about different bioinformatics options as well as computational biological methods. So last year, when the COVID pandemic hit, to support the NIAID COVID-19 consortium researchers, we reached out to LabKey to basically be able to view it as a platform to be able to integrate their data for their study for the immune response to the COVID-19 study. So this was last year, and we also had a presentation to talk about how our progress was with that study. To date, we are actually presenting to you the extension of that same framework, where now we are able to support other labs under the Division of Intramural Research. And one of the studies that I'm wanting to talk to you about today is about the under the Laboratory of Clinical Immunology and Biomicrobiology. The study is basically called as the Transitional Autoinflammatory Diseases Unit. So this study and this research team is basically focusing on rare autoinflammatory diseases. So in this effort, they actually collaborate with a lot of specialists across NIH, where they're actually sharing information by providing high throughput technologies, as well as their research and clinical tools to be able to evaluate these patients that are coming across. So as I build up the study, or build up the information about the study, we are not strictly just looking at only the samples aspect of it, but also trying to find a platform which was going to be able to integrate studies of, data coming in different types, So in the next slide, as I'm going to show you, before we were thinking about LabKey, this particular study was having multiple protocols which were actually storing their data in multiple resources. So all these different data types that you're seeing here, which is the clinical data, the genetics data, the lab data, as well as the samples that you see, all of this is a part of the study. So traditionally, the team was basically trying to identify this information by making sure that they were downloading all this information in multiple spreadsheets, and as you can kind of gauge through, it is a lot of effort and a lot of work and a lot of meticulous work that the user actually had to make sure to make, kind of end up with their analysis for these rare diseases and studies. So some of these clinical systems are research information systems, are within NIH and NIAID, but even then, you can imagine that it's these individual spreadsheets that are being downloaded, then tracked, and making sure that it's the same patient that you're looking at this information across. So you can kind of understand, or kind of gauge the process of it being really laborious, as well as it having a lot more chance to have manual errors, and it could make the process inefficient, even though the team was really meticulous about maintaining all these spreadsheets. Something to what Jess had just referred to. Imagine the number of versions of spreadsheets that they would have to look through in order to come up with their analysis and efforts. So once LabKey came into the picture, this is what our effort or our aim has been in terms of addressing these challenges, which is we are able to now download the data from all these individual clinical repositories into one single platform, where the researchers now would be able to look at information across their longitudinal data, because they're getting a view of, kind of getting the cross-section of all their subjects and their conditions right across in one location. They're able to query the data sets across as well to, come up with their own analysis or come up with their own research questions, as well as be able to kind of use this research data and then collaborate with other users in other platforms. So this is basically kind of showing the engagement of NIH in not just collecting the data, running their own research, but also sharing this information with other researchers to kind of continue or accelerate the discoveries that could be made in this field. So, so one of the key things in this case was we were not just looking at using Sample Manager as on its own because of the nature of the data that was being captured for this group, but we are also looking to give them a view of where they would be able to combine the data that they are being able to see for their clinical aspect, for their lab aspect, as well as the genomics aspect that was coming in, along with the samples that was being collected within their study. So we were looking to kind of reap the benefits of using the LabKey server and the study model within there, along with the LabKey Sample Manager to be able to track the samples. So one of the examples that we would like to see in here is that we would like to see the, kind of see the demographics of a particular patient and their diagnosis and treatment that was being provided for them, along with the samples and the genomic aspect that was being collected. So it gives you a quick snapshot of that one single patient by just navigating between these two systems, or by coming up with queries and information that we would be able to assign, be it different queries, be it different reports that could be generated by either using the ingrown out-of-box features within LabKey itself, or create reports that could look across this information across these two, across the server as well as the sample manager. One of the key requirements of this group was also to be able to share this information with an external platform called as Annual, or the NIAID Genomic Data Science Analysis, Visualization, and Informatics Lab space. So one of the things that we are focusing on in here is to basically create workflows that would help query this information and package this information for those platforms in a standardized and harmonized fashion. So, basically the data that's coming in is being shared so that the information that you're looking at would be the same, would look across in a similar fashion. So having said so, in the sample manager, what we're basically focusing on is having the ability to track the longitudinal samples that is going across the study. So one thing that we had to be mindful about is that this is a study that's still ongoing. So we have data and samples that is collected from, from prior collections, which is still there on their spreadsheet. So it's, being able to address their retrospective samples as well as the prospective samples that was going to be coming in. The reason being that we want this process to be as seamless as possible for the researchers as they would like to integrate their clinical lab information along with the research data, and make sure that we would be able to come up with a kind of a standardized format in order to have this tracking in place. So, excuse me. So like I mentioned, since we are having all this data coming in from this multiple sources, it's really essential to create a unique identifier that would basically connect all these connect the patient across these different data sets, as well as continue that into the sample manager, because of which we are trying to incorporate that into the naming convention, something that Hannah was just mentioning, where it basically allows the lab to kind of recognize their samples and continue to do the same as they progress to the prospective samples, even when they're using sample manager. So it helps then, kind of continue that lab workflow as well as manage the new samples that's coming in. So there's no confusion about, is this an old sample? Is this a new sample? Are we overlooking something? So it basically helps them focus on, the diagnosis that they would like to come across with a quick look, by using LabKey as a one-stop shop. So some of the examples that I'm showing in here is basically how we were trying to come up with a standard naming convention by using the enrollment ID, the data collection, the tube types that is there. This is really something that the team can relate to and connect with, and LabKey is giving us the flexibility to come up with those naming conventions. So in terms of the features of the LabKey that we're trying to look in, especially with the sample manager, is We are interested in, of course, tracking the samples for both the retrospective as well as the prospective samples coming in. We are interested in maintaining the lineage of the samples that's being looked at. And in future, something that Hannah just referred to, the group is also interested in looking at how they would be able to incorporate barcoding into tracking their samples that's coming in, as well as using the freezer management in order to track all these samples, both retrospective as well as the prospective samples that's going to be coming in. So all of this would not have been possible without, kind of input and guidance from Hannah and the team in order to be able to, one, understand the requirements that we were receiving from the labs, to understand their, the magnitude of the data that they had and how we would be able to kind of, make it as efficient as possible for the teams so that they would be able to focus on the research aspects. So we do continue to refine and streamline those data workflows, working with the team and trying to make sure that we would have the system up and ready for the team to kind of use. So that's where we are at right now. And we are open to any questions if there are any. Thank you.
Thank you so much, Smilee. I'll do the same thing here. I thought that was a great presentation. I've got my own question to start while we wait for others to sort of get theirs. But it's a little sort of tangential to just sample manager, right? You also talked about study and the integration of that clinical data with the sample data. I know how you guys decided to implement this, and because I'm your account manager as well, but very, very curious, if you guys had the option to start from scratch or do this again, where would you start in terms of what piece components would you start with first?
So in this case, I think we would use the same model that we used for this particular study, which is, starting off with the clinical aspect of it. So we started off in the server within LabKey server first, using the study model. The reason being that, it was really key to identify what would be the key, kind of the unique identifiers that would connect them across, right? So in this case, we were able to identify that following through with the study's ID was the best thing that would help us identify all these, data sets going across, because being mindful of the fact that we are connecting to all these different systems, which have their own IDs and identifiers. So, it was really key and essential to, kind of direct the scripts and the APIs to kind of hone in on that one piece and then move on. And so moving from there on to the sample manager then became easy, because we are now using the enrollment ID as one of the parts of the naming convention, which kind of helps the users to continue with what they're being used to. So
awesome. Okay, I think that's it. We're no questions here. Oh, one more came in. Smilee, how is your demographic data feeding into sample manager from study?
So we are connecting to be one of NIAID systems to be able to pull in the demographic information that's being collected at the clinical center. So, it's basically the enter that was seen, that has been entered at the clinical visit that is being pulled into the system. So it's not directly being entered into LabKey, but we are connecting to that system to pull it in.
Awesome.
All right.
Thank you so much.